You guys hear me? All right, good. Yeah. Well, thanks for coming out so early. It's always a pleasure to be back here at EMSL. I've collaborated with EMSL for many years and published a lot of papers. I'm always very appreciative of the work and expertise and all the technology that's available. Oh, gee, get rid of that. Okay. So today I'm going to talk to you about some work we've been doing on plant receptors. And this is in, of course, conjunction with EMSL, as the title indicates. One of the things to keep in mind is that in Arabidopsis and plants in general, very different from humans, for instance, there's an enormous number of receptors. And you can put this down to the fact that plants are stuck in place. They can't run away from danger. And therefore, they need a lot of receptors to be able to respond to their environment. Now, of the 600 receptors in Arabidopsis and roughly in tomato, and there's over 700 in soybean, which is another plant that I work on, we know maybe the function of maybe 10% of these. And so the question then is, what do the others do? How do they function? What determines their specificity? So on and so forth. And those are the kinds of questions that my lab is interested in. Now, plant receptors, the kinase domains of plant receptors look to be monophyletic. But the extracellular domain is what really classifies these. And so they have a variety of different extracellular domains, which presumably bind to various ligands to carry out the signaling function. Now, some years ago, we got involved in studying extracellular ATP. And extracellular ATP, the analogy I use is, let's assume you have $1,000 in your pocket and you're walking down the street, and every so often you throw a dollar bill over your shoulder. People see you doing that, they would probably think that you're crazy. But on the other side of that, pretty soon you'd probably have a big crowd of people behind you, following you, picking up these dollar bills. And so it doesn't really make sense that plant cells or human cells would actually excrete ATP. But at the same time, when you do that, of course, you're going to get attention. It's going to be an important signal. And so there's a guy named Jeffrey Bernstock who is dead now. He died in his 90s, but he was a professor at the College of London. And he started studying extracellular ATP back in the 70s. One of the first observations was, if you add extracellular ATP, it causes muscle contraction. But for a number of years, he was really ostracized. I often tell my students, they say, how many of you would like to make a paradigm shifting discovery? And students, of course, most of them just look at me, don't do anything. But there's always a few brave souls that raise their hand. And I say, be careful what you wish for, because science is a conservative community. And if you really make a paradigm shifting discovery, you're not going to get patted on the head. You're probably going to get hit in the jaw. And this is exactly what happened to Jeffrey Bernstock. So he would give these talks at the meetings I would go to, and he was extremely bitter about how he was treated early on. But what happened in the 90s, early 90s, is the first receptors for extracellular ATP were identified. And once people realized that there were actually receptors for extracellular ATP, people said, maybe the stuff that Bernstock's been talking about is actually real. And so you see then there was an explosion of papers after the 90s. And now we know that ATP, of course, is the energy currency of the cell, but when it's extracellular, it acts as a signal. And we know that in mammals, extracellular ATP plays a variety of roles, including in neurotransmission. And so as the vesicles fuse at the side of the neuron and release neurotransmitters, one of the transmitters that they release is actually extracellular ATP. And so there is now a multi-billion-dollar drug industry around extracellular ATP. And a few of you may be old enough to know what Plavix is. Plavix is a drug that if you ever have a heart attack or a stroke and you go into the hospital, they'll give you Plavix. And what Plavix is an inhibitor of the P2Y12 receptor involved in clotting. And that drug alone, it's off patent now, but before it was on patent, it was $9 billion in sales per year. And it targets one of the types of receptors that exist in mammalian cells, this P2Y receptor, which are G protein couple receptors. The other type of receptor in mammalian cells is P2X, and these are ion channels. Now, plants don't have P2X or P2Y receptors. But we know that there's a variety of papers showing that if you add extracellular ATP, to plants, you do see some kind of response. So it was known for quite a long time that plants respond to extracellular ATP. And one of the things is ATP in humans and also in plants is what we call a damage-associated molecular pattern, DAMP. And so when the plant is damaged, it releases ATP, just like we do when we cut ourselves. And that ATP then is recognized by receptors and activates a response inside the plant cell. So knowing that, and this shows wounding, so this is some transcription stuff that we did some years ago, showing that there's a significant overlap between the transcriptional response to ATP and the transcriptional response to wounding. And the same is true of stress. And so again, this is from a review paper we wrote. So we see there's a overlap in sodium chloride, UV, and a variety of other kinds of stresses. And so now what we know is that when a stress comes in, be it damage or a pathogen or whatever, one of the things that happens is the release of ATP, and that then is recognized by the receptors and is involved in this stress response. And this just shows you using luciferase, which is an enzyme that produces light and is dependent upon ATP. When we add luciferase, these are root hairs. Here's untreated. These are root hairs that are treated with chitin, which is an elicitor. And you see a very, very strong extracellular ATP release when you add things like chitin. So well, I guess it's 13 years ago now, this was kind of our understanding of extracellular ATP reception. So we knew of these receptors in animal systems. We knew based on sequence comparison that P2X, P2Y receptors were absent from plants. And so the question then was really, what is the receptor in plants? And knowing the history from the Bernstock story that I told you, it became obvious that we could do a lot of stuff with extracellular ATP, but the community in general was not really going to accept anything that we talked about until we identified what the receptor was. And so Kiwamu Tanaka, who's now a professor at Washington State University, a few miles up the road, and Jungmin Choi, a PhD student in the lab, did a mutant screen. Jungmin has her own lab now at the University of Cambridge, actually did a mutant screen, and out of that, we identified a receptor called LECRK or DORN1. DORN1 says doesn't respond to nucleotides, which is the screen. So basically, we screen for mutants that would not show a calcium response in response to ATP. But since we have P2X and P2Y receptors, when I started giving these talks in the animal community, they said, that's a strange name. These are P2 receptors. And so we've kind of adopted now this name P2K, the K meaning that it's a kinase. And so this receptor has an extracellular lectin domain, which binds ATP at a relatively high affinity, shown here, nanomolar levels, a single transmembrane domain, and then an intracellular serine threonine kinase domain. And out of this, then, Jungmin won the outstanding PhD dissertation for the entire United States. So it turns out that I had no idea about this. I learned about it because of this, that if you, at the university, if you have, at our university, I think all universities, when you submit your dissertation, it goes into a competition for the best dissertation at the university, which she won. And then the best dissertation at the university is entered into this national competition, and she won the national competition. So they flew her from Cambridge over to Seattle, gave her a check, I think, for $4,000 and a nice plaque and a nice picture. And I think I was in Brazil or something at the time, so I didn't get to go. But anyway, so Jungmin got that, and she's done very well since. Okay, so now we know that there, as I said, there's multiple receptors in mammalian cells. And so far, we've identified two receptors, the original one, P2K1, and another one which we call P2K2, and they interact to form a complex. And now, through a whole variety of papers, we've really started to understand how the signaling takes place and the downstream effects, and I don't have time to go through all of that. But these are the kinds of things. And so in addition to acting as a damp, we know that it controls what we call stomatal immunity, the ability of the bacteria to infect through stomates. It activates ROS, secondary metabolism. Papers that we're writing now, it controls cell death and controls plant growth. I'll talk to you a little bit about protein acylation, controls sensitivity to geosmonic acid, calcium influx, which of course is the assay that we originally used, and so on and so forth. So a lot of activities are tied to these receptors. And this is an example. This was a paper we had back in 2017 where it controls stomatal immunity. And so ATP actually closes stomates, and the Pseudomonas syringae that we heard Anich talk about the other day actually infects through stomates. And so ATP closes the stomates, and by so doing, makes the plant more resistant to the pathogen. Okay, now the other thing more recently we've done is you may not be aware of it, but the plants actually communicate systemically. And so if you treat one leaf with a particular stress, be highlight, cold, wounding, whatever, there's a signal that goes and actually tells these adjacent leaves that there's some stress occurring. And so these adjacent leaves then become resistant. And so there's even though you're just treating one leaf, you actually see a systemic response. And one of the things that happens is a so-called Ross wave. And so if you put a localized stress on one cell, then reactive oxygen species is produced, and then that then can be transmitted through the plant, and again, part of this systemic response. And so this is a movie we have. This is work we did with Ron Mittler at our university. And so in this is a control plant. This is a plant where we've wounded this particular leaf, but these leaves are unwounded. And again, you're going to see, and we're looking at Ross here, if the movie works. Yeah. So you'll see that the control plant, nothing, but you see that there's been this Ross wave that moves across the plant. And Ron's done a lot of work on this Ross wave. And then the other thing we do is if we look at that leaf and wound it and look at the ATP coming out of it, again, using luciferase, we see there's roughly somewhere between 20 and 45 nanomolar ATP that's actually being released from the wound. And then we also showed that, again, this is the 0 time and then wounding. Here's the control. Here's we've added, and you can see the Ross wave moving along. And if we do the same thing in the P2K1 mutant, you don't see the Ross wave. So these receptors are responsible for recognizing ATP and then triggering their Ross wave. And this is a model for this. And so you've got wounding, releasing ATP, activating this, and then activating things that are calcium channels and production of Ross, and so on and so forth. So an example of some of the kinds of things that we've been looking at. Now, what I really wanted to talk to you about is this acylation. So it turns out that, and this is at EMSL where they have a lot of mass spec capabilities. And so here at EMSL, you can measure all of these covalent modifications. And of course, things like phosphorylation have been studied very broadly, but there's a variety of other kind of covalent modifications that occur to proteins that are less well studied. And one of them is this idea of adding a lipid group, acylation, or sometimes called pamidylation. And that's what I want to talk to you about. And so these proteins that exist in the membrane, some of these are protein acyltransferases, PATs, and they then add a palmitoyl group. And so sometimes this is called pamidylation, sometimes it's just called acylation. And of course, that will make the proteins more hydrophobic. And it's reversible. Then there are other enzymes that can actually remove that acyl group. And in mammalian cells, where it's been studied a lot, it's really involved in moving and targeting proteins to the membrane. And so these proteins become acylated in the Golgi. That then targets them to the membrane, where they can associate with the membrane. And of course, when it's cleaved, then they can recycle back and be degraded or turned over or whatever. Yeah. Now, in mammals, there's only 2 of these acyltransferases that are found on the plasma membrane. There's several of these in mammals, but there's only 2 that are found in the plasma membrane. But if you look over here in plants, you see that roughly half of all the acyltransferases that exist in plants, this is Arabidopsis, are actually found on the plasma membrane. And that may in fact be an indication of the large number of these quantify this, we find out that that acylation is necessary for these things to co-localize in these nanodomains. So there's a role for this acylation and localizing these proteins to the nanodomains. Now then, obviously, you know, this is based on confocal microscopy, and the images are okay. When we tried to publish this work, we got heavily criticized. People were not that convinced by what we were saying. So, you know, that's the way it goes. Now, again, there's all of these different PAMP receptors. So all of these receptors are involved in pathogen recognition. So the question then is that in the Cyril Ziffel paper, they looked at two proteins, similar structure, but different function, found them in different nanodomains. Now, if you have proteins that have the same function, that is PAMP recognition, will they be present in different nanodomains, or will they be present in the same nanodomain so that maybe you have like a pathogen response nanodomain? And so with De Hong's group, then we decided to use single molecule resolution storm microscopy to actually look at these, to answer the criticisms of the reviewers, but also to get a better view. And this is work that was done by Jeremiah and his group. And so here just shows one of these things, and wherever you see these dots, this is based on fluorescence. You can see the localization there. And so this is a paper that we published this year. And one of the things that came about is we know that when we add the ligand, and this has been known for a long time, that when you add the ligand, the receptors internalize as a way of shutting down the response, because you don't want the activation to go on for a long time. And so basically he looked then at basically the turnover and also the co-localization between remorin, which was a known nanodomain protein, and FLS2, the flagellin receptor. And sure enough, that when you add the ligand at time 0, there is a reduction in the density of the receptor on the membrane, and then after about 90 minutes, that gets restored, which again is stuff that was known. What came out of this interesting, though, is since we had remorin labeled and FLS2 labeled, we could follow the dynamics of both of them at the same time. And what came about from that, and this is a diagram from that paper, is that when FLS2 is internalized and remorin is internalized, and it's known, there's a paper showing that remorin is necessary for that internalization. But as they come back into the membrane, they come back independently. And so FLS2 and remorin do not come back and immediately co-localize. They actually come back and then later on have this association. And that's what this model shows and what his microscopy showed. Now, so that was that paper. And then we also then wanted to look at these other receptors. And so again, we were looking at the flagellin receptor, the ATP receptor, and the chitin receptor. And again, this was on root hairs, looking at the localization of this. And we're asking the question again, this co-localization, are these three PAMP receptors all found in the same nanodomain? And the answer is no. So at most, about 20% of the receptors co-localize. And there's more co-localization between FLS2 and the ATP receptor than there is between the chitin receptor and FLS2. So now that 20% that localizes could be important. So an open question is whether or not that 20% that co-localizes, or at least appear to co-localize, is that playing some kind of unique function? But in general, 80% of the receptor there, they are not co-localized. And so again, just as in the case of the BRY1 and the FLS2 story I told you from Cyril's lab, it appears that these receptors, even though they're PAMP receptors, or all PAMP receptors, are found in unique nanodomains. Now, remember back, there's 600 receptors in Arabidopsis. Does that mean there are 600 unique nanodomains in Arabidopsis? So each of these receptors has its own unique nanodomain. And if that's the case, that may explain a lot of the specificity. So I told you in BRY1 and FLS2, they have similar structure, they have similar cofactors. So what determines the specificity of that receptor? Clearly, the specificity of ligand binding is important, but perhaps also the fact the environment in which that receptor finds itself within the nanodomain could also be a very important determinant in how the specificity of that receptor functions, not only in ligand activation, but in activation of downstream events. Okay, so more recently then, Jeremiah and De Hong have used the latest light sheet, and he's here to answer any questions of this because it's beyond me. But the beauty of all this, of course, again, you can do single molecule measurements. And so again, using these, and here's the picture that I wanted a picture with Jeremiah in front of it. I asked him for it, but he never sent it to me. So imagine Jeremiah standing there. So here's a picture that Jeremiah sent me, and this is a movie. And so the blue represents FLS2, the green represents P2K1, and the red circles represent co-localization within about 500 nanometers. And based on Jeremiah's measurements, these nanodomains are somewhere between 300 and 500 nanometers, which also matches what's known in the literature. And so you can see here the green dots and the blue dots, and there's relatively few red dots. And again, I told you that his measurements indicated maybe at most 20% of these receptors co-localize. By and large, they don't co-localize. And this just shows you the light sheet picture, gives you kind of an orientation of how the tissue is there. And again, the green is FLS2 and the red is P2K1. And this is another picture. The other thing he can do here is he can look at where the membrane is and then where the receptors are. And again, this is important from the standpoint of internalization. So one of the measurements he's made is the distance between where the receptor is and where the membrane is. And of course, as the protein gets turned over, the distance from the membrane gets larger. And so those are the kind of measurements that he's made. And that shows you here. So here is the absence of ATP, and then when you add ATP, the distance from the membrane increases because we know that adding the ligand causes a turnover of the receptor. And again, and he can actually calculate the distance from the membrane when that happens, and look at the internalization that takes place. And again, there's very, very little co-localization that's taking place in this particular case. Now, this was all very interesting, but what was, so here's also the nearest neighbor distance. Again, these things are within about 500 nanometers and at most about 20% of the time. So again, there's very little co-localization between these receptors. Now, what was exciting to us, though, is this picture. So, and this is my last slide. So over here is looking at P2K1 FLS2, and again, the different colors, and red shows co-localization. And this is, again, the localization of P2K1 FLS2 when you add ATP. Now, FLS2, reminding you, FLS2 binds flagellin. It doesn't bind ATP. But notice that here are the patterns that exist between P2K1 and FLS2. And then when you're adding ATP, notice that there is a redistribution that's taking place, and a redistribution of P2K1 more similar to auxin and cytokinin, because it's playing a variety of roles, not just in a stress response, but it's also playing an important role in growth, cell death, development, and so on and so forth. So I actually think of ATP as more like a hormone than a stress response, although I think the plant physiology community is still catching up with me. As in the case of animals, plant receptors are localized within these membrane nanodomains, which have distinct lipid composition and protein composition. And we're now doing studies with EMSL to actually define that lipid and protein composition. Plants have massively expanded repertoire of receptor-like kinases, reflecting their stationary lifestyle and need to respond to an environment. Available data suggests that distinct receptors primarily occupy distinct nanodomains. So again, if there's 610 receptors, are there 610 unique nanodomains? And perhaps contributing their specificity, but raising the interesting question, given the large number of receptors. Ligand binding does not appear to change localization within nanodomains, but it does trigger receptor internalization, which is not something we discovered, but something that was known. And then finally, perhaps the most interesting, extracellular ATP in addition drastically changes the distribution of both P2K1, but also FLS2 nanodomains within the plant membrane, suggesting that ATP addition may also affect the plant response to other elicitors. And this, again, are experiments that we're doing right now. So again, I probably went too quickly, but here's the group. And thanks to all the collaborators, many of which are here at EMSL. Been a very productive collaboration. Probably over the last 15 years, we've published 30 papers or something like that. So it's been a very, very good experience for me. It's always fun to come back here for no other reason than to drink the wine. So thank you very much.
We have time for questions. While Heather's walking over there, a question: what do you think all the reactive oxygen species are? Like, what kind of signaling do you think is actually happening when you were showing some of those earlier videos and stuff like that? What kind of molecules do you think they are?
So the idea behind the ROS signaling is that the ROS is actually not in a way moving. So what's happening is the ROS is being produced by one cell and then triggering a response in the adjacent cell, which is causing ROS to be released. And then the adjacent cell is causing it. So it's a cascade. It's not really a movement of ROS that's taking place. And that's similar. So some of you know Ryan, Dr. Ryan, that used to be at I think Washington State or University of Washington. He had this peptide. And again, initially the idea was the peptide was moving the plant, but then later it was discovered that again, it was this cascade that the peptide would trigger a response, and then that would release the peptide, which would trigger a response. And so it's not necessarily the ROS moving multiple cells, it's just that they're triggering this cascade of response.
Gary, excellent talk. So I was wondering, I don't know if you mentioned at all any autophosphorylation or phosphorylation events that taking place as part of the internalization process of P2K1?
Yeah, so we've studied that a lot and we published several papers on it. Yeah, so P2K1 autophosphorylates and then it transphosphorylates. And we've shown with kinase dead versions that phosphorylation is essential for signaling. And we've identified a whole variety of targets for the kinase. One of the interesting things, if you don't work on protein phosphorylation, very often you see these papers where people will study a kinase and they'll identify a single protein that's a substrate and then publish a paper in Nature or something and then so you come away with the impression that each kinase phosphorylates one protein. Well, that's not the case. I mean, each kinase phosphorylates multiple proteins. And that slide, my colleague, Jay Thalen, who some of you may know, developed this assay called the KICK assay for kinase client. And so what he did is he developed a library initially of 2,000 peptides. These were generated from known phosphorylation sites in Arabidopsis. There are 20 amino acid peptides, 2,000 of them. And what you do is you purify your kinase, and then you add it to this mixture of peptides, allow them to phosphorylate the peptides in vitro, and then load it all on a mass spec to identify which of the peptides are phosphorylated. And so we've done that for all of our kinases that we study. And we find that roughly 50% of those that we can confirm by other experiments. It may be that the other 50% are also real, but we can only confirm half of those. But even so, even U2 hybrid, 50% success rate is about the norm. And so now with Jay, we've expanded that 2000 library up to 8,000. So we have a library now of 8,000 peptides that we can screen with these kinases. And he's open to collaboration. So if any of you have kinases, it's a very effective way to identify what the substrates are for your specific kinase. That was probably more than you wanted to know, but yeah.
That's good. I'm a kinase guy. And then, so the other question I had, that what is the dynamics of, the calcium influx versus the acylation, deacylation, as well as the internalization?
Yeah, so it's complex, and this was one of the problems we had in publishing the paper. So there's a dynamic between acylation, which is inactivating the protein, and then autophosphorylation and transphosphorylation, which requires the acyl group to be removed, and then internalization. And so it's a very dynamic thing, and it's happening in minutes. And so you've got acylation, you've got removal of the acylation, you've got autophosphorylation, and then of course you have turnover of the receptor all happening at the same time. And it's something we'd like to study, but it's a little bit beyond us right now.
Really fascinating talk. I was wondering how you're thinking about what is defining that localization to those nanodomains. Is that based on protein interactions, or is that those acetylation lipids?
Well, it's clear that the acylation is important, but it's also clear that there are important protein interactions there. And one of the things that we're able to do now, and would like to do this work with EMSL, although we've been recently stymied, as Chris knows, is we can now, using epitope-labeled receptor, we can actually immunoprecipitate nanodomains for each specific receptor. So we can look at the composition of the nanodomains in general, and then we can immunoprecipitate and enrich for only those nanodomains that contain a specific receptor. And then hopefully using lipidomics and proteomics, we can go in and look to see how the composition of the receptor-specific nanodomains is different from the general population. And we're hoping that those kinds of experiments will get at some of the questions that you're raising.
Okay, one last question, please.
It's a fascinating talk. I just want to go a little bit deeper about both the concentration as well as spatial distribution of the ATP, and how that related to this dynamics.
Yeah, so we've done, one of the early papers that we published was with Alan Jones at the University of North Carolina. And he showed that if you go in and you take a needle and you just barely touch the plant with a needle, you release nanomolar levels of ATP. So just touching the plant causes ATP to be released. As I remember, it was 10 nanomolar or something like that. And then I just showed you here that based on this wounding that we did, we can get maybe 50 nanomolar released. And then other experiments that Stan Rue has done at the University of Texas, if you wound the plant and then allow the liquid to kind of accumulate in the wound, he's been able to measure like 30 micromolar in the wound that's accumulated over time. As I told you, the receptor has like 20 nanomolar, that's the Ki, or the KD for ATP. So those are all within the physiological range. Yeah. Thank you.
All right, let's thank our speaker one more time.
Our next speaker will be Professor Ziva Kamas from Florida Atlantic University. He will talk in remote. His title is Mapping Microbial Biogenetic Gas Activities in Peat Soils at Multiple Scales of Measurement Using